Okay, so let's get going. First of all, what's in this webinar? I'm going to introduce Kirkstall as a company, then I'm going to talk about the Quasi Vivo concept, moving on to our systems, and then looking at three of our major applications: liver, cardio, and co-culture. And finally, there will be the opportunity for a question and answer session at the end. So, Kirkstall's mission. Our tagline is saving lives through better science. And we hope that our Quasi Vivo system can deliver wide benefits to the research community to provide all of you scientists with the tools and the equipment that will allow you to precisely control the environment for the culture of human cells. And we believe this will significantly improve the human relevance, robustness, and repeatability of biological research. We're a small company. We're based near Sheffield in the UK. So, the concept. The system itself was invented by Professor Arti Ahluwalia and her team at the University of Pisa in Italy. And you can see this diagram in their thought process in the paper that's quoted on the screen here. But basically, they were looking at the different kinds of stimuli that traditional cell culture seems to miss. They first started looking at biochemical stimuli, so things like hormones, cytokines, and growth factors. And as we know, we can provide all of these kind of things in the cell culture medium. We can also start mimicking the structural stimuli that cells require, so different architectures in a 3D environment, by providing different 3D structures for cells to be cultured on, such as scaffolds and gels. Especially if we're looking at the use of gels, we can start to modify the stiffness and the viscoelasticity of the environment. The one bit of the internal environment that static culture misses are all the kind of fluxes that cells in our bodies experience. So changes in energy and the different momentum and flow stresses that cells do experience in vivo. If we can start to apply all of these different things to cell culture, we can significantly improve the human relevance of our cell culture systems. We think that all cells are exposed to flow. So in the Quasi Vivo system, we're not really designed to mimic the sort of high pressure, high speed blood flow that we see within the circulatory system. And that's shown in this diagram at the top. What we mimic using our system is the much more gentle interstitial flow that comes out of the circulatory system and into the cells and tissues. This is the kind of flow that the Quasi Vivo system was designed to mimic. What are the benefits of flow within the in vitro environment? Well, we can deliver oxygen and fresh nutrient to the cells that are cultured within our chambers. At the same time, we remove and dilute waste products into the flow. We believe that the flow itself is stimulating the cells. We think they enjoy being a little bit stressed out by a little bit of flow application to the cell culture. We also believe that flow provides a better mechanism for cell-to-cell signaling, and eventually this will enable us to start building systemic and multi-organ models. This just basically allows the cells within the system to talk to each other. And here's a diagram of the Quasi Vivo chamber. So this is the Quasi Vivo 500. When it was designed, there were extensive computer models done on the system to ensure that the flow was adequate and the oxygen conditions at the base of the chamber were adequate to allow cells to thrive. So here you can see on the left-hand side is the inlet, and the right-hand side we have the outlet. There are two things that you might notice from this diagram. The first is that the inlet is both lower and narrower than the outlet. And the second thing is that the roof itself is sloped towards the direction of the outlet. This is to ensure that we don't get any bubbles within our system. And if we do get bubbles, they're quickly shepherded from the inlet towards the outlet. Within the chamber, the flow is laminar up to pump speeds of 1 ml per minute. But if we look at the flow characteristics within the chamber, the flow rate as delivered by the pump is only seen at the very top of the chamber. As we move down the chamber towards the area at which the cells are cultured, the flow rate is around 300 times lower than it is at the top. And this means that we achieve physiologically relevant shear stress and velocity peak levels. You can read all about the development of our chamber in Mattei et al., and that's the paper quoted on this slide. And here's a diagrammatic representation of a Quasi Vivo circuit. So here we've got two different circuits, one on the top and one on the bottom. We have a reservoir bottle. This provides enough media for the cells for around 3 to 7 days. This depends how metabolically active your cell culture is. Obviously, if it's more metabolically active, it's going to use the nutrients up faster, so the media will need replacing more often. The flow itself is recirculating, so it comes out of the media bottle, goes around the system, and then dips back in. This allows cells to condition the media, so they're really creating their own environment through the expression of different proteins, so that they're really mimicking what happens within the body. These examples have 3 individual cell culture chambers per circuit. The current record for chambers in a row is 12, but the most common we find with our users is 3. In this example, we've got a scaffold being held in the base of the QV500 chamber, but you can grow your cells on cover slips, in gels, and we've even had some users directly placing tissue slices inside the system. This example shows 2 circuits. We do have a simple peristaltic pump that provides controlled flow in 2 channels. We also have a more complicated version, which will do 6 channels at the same time. But basically, the system is designed through the use of Luer locks and standard tubing, so you can connect it up to any kind of peristaltic pump that you might have in your lab. So that's what the Quasi Vivo system looks like in concept form. And these are the 3 different chambers that we have. So our original chamber is the QV500, and then we developed the QV600, and finally the QV900. And I'm going to go into each of these chamber types in more detail now. So the QV500 was the original chamber that came out of the University of Pisa. As I said, it's designed to eliminate bubbles and to provide laminar flow. The whole system is fully submerged in liquid, so it allows for fully submerged cell culture. It has a modular nature, so you can do interconnected cell culture. So you can, as I said, link 3 together, you can just have 2, whatever experimental design you require. And in the table here, we've just got some details about the material of the chamber, how big it is, and what it's made out of, for example. And you can see at the bottom picture here, the chamber consists of a base, which is where the cells are cultured, and a top, which is where the tubing is connected. And these 2 halves of the chamber lock together really easily, and then the whole system is completely sealed. In the picture on the top, you can see an example circuit with 3 chambers in a row, powered by our 2-head pump with a reservoir bottle containing the cell media. And the volume of this chamber is 2 mils. The QV600 was designed when we were talking to some of our users, and they really wanted to do air-liquid interface culture in our system. So they were interested originally in skin, but also in respiratory models. And they wanted to be able to drop a commercially available transwell with their cell culture inside the system, so that they could expose the base of the transwell to flow and have the open exposed to air. So we had a look at designing this, and this is what we came up with, the QV600. So it allows for the creation of initially this air-liquid interface. And this is through just dropping in commercially available transwells. And here again is a table showing you all of the dimensions and the materials that this chamber is made from. When we started talking to users and showing them our chambers, we had a couple of comments that suggested that we could also use it for liquid-liquid barrier models. So things like gut models, blood-brain barrier, that kind of thing. So we slightly modified the chamber. So at the bottom picture here, you can see an insert with an O-ring around it and a plunger from a syringe. And we can use these 2 pieces of equipment to place the barrier between the top 2 tubes and the bottom 2 tubes. You can then culture your cells on the top of this membrane, on the base of the membrane, or both, and expose both sides of the insert to flow. With the lid on, then, again, as in the 500, the whole system is completely sealed and it's sterile. In the top picture, you can see the 2 different versions of the QV600 running. So on the left-hand side is an air-liquid interface example. So you can see the base of the chamber has media and the top does not. In the example on the right-hand side with the blue and the pink liquid, you can see that we're running it in liquid-liquid mode, which allows for two different flows to be applied at the top and the bottom of the chamber. The volume of this chamber is 2 mils when you're running it as an ALI, and obviously 2 times 2 mils with your barrier insert. Finally, we have the QV900. So this was designed after talking to some of our industrial customers who wanted, first of all, a slightly more high throughput version of the QV system, and they also wanted it to be optically clear at the base so that you could place it onto a microscope just to have a look at how your cells are doing, or to do any different kind of endpoint assays that require you to actually see your cells within the chamber. So we're designing this as an organ on a plate disposable unit. So it's ideal for users who require more high content, high throughput experiments. On the right-hand side table here, we've got some details about the chambers themselves. The main difference between the 900 and the 500 and 600 is the material that it's made from. In order to make it optically clear, we had to choose an acrylic plastic rather than the silicone, which is what the 500 and the 600 are made from. This means that it's one use only, so you can't autoclave and sterilize this chamber, whereas the 500 and the 600 can be run through the autoclave a few times, and they can also be sterilized with alcohol. The volume of the QV900 is around 4 mils. And here as well, you can see an example of a 6-head pump. So in this particular experiment, we're running the chambers in sets of 3. So 3 chambers connected to one reservoir bottle and 1 pump head. So in this example, we've got 6 different circuits. So for example, if you were doing some compound testing in the lab, you could have 5 different concentrations and your control. So if we continue to think about the more industrial questions that Quasi Vivo might help to answer, and if we look at the drug development pipeline, we're moving from drug discovery, which has thousands of compounds, through preclinical research, preclinical trials, and then into humans, validation and commercialization. And on the diagram here, I've indicated where I think Quasi Vivo sits within this pipeline. So with Quasi Vivo, we have high content assays that are relevant to the clinic, and we're increasing our confidence in the results that come out of our in vitro tests. Initially, we believe that Quasi Vivo sits either before or alongside in vivo animal trials. Beforehand, this means that we can reduce and refine the animal experiments that we're doing by giving more confidence in the compounds that are going into in vivo trials, and alongside them, so that we can validate the data coming out of Quasi Vivo with the animal models. Obviously, eventually, our aim is to completely replace animal models with our Quasi Vivo system. We've got a number of existing users using Quasi Vivo technology throughout the world, so used in the USA, in the UK, and across Europe as well. We have a number of published papers. You can see these on our website, and there's also a list at the end of the webinar. So the system itself was designed for hepatocyte cell culture, so the main bulk of our data is on liver cells. We do have some data available on stem cells, kidney, cardiovascular, fibroblasts, and diabetes models. However, we've got a number of users who are still in the pre-publication stage. They're still busy working away using the Quasi Vivo system. So these users are looking at things like different cancer models, skin models. We've got some people looking at gut, brain organoids, blood-brain barrier, and respiratory. Basically, the Quasi Vivo can be used for any kind of cell culture that you're interested in. The first application we're going to be looking at in today's webinar is the lung. And this data was recently published in a journal in Scientific Reports by some of our collaborators at the University of Innsbruck in Austria. And they developed this lung model using normal human bronchial or small airway epithelial cells from Lonza, cultured at the air-liquid interface in the QV600. They exposed the cells to perfusion or to static conditions, and they found that perfusion resulted in a significantly accelerated development of the lung epithelia. As you can see in the top panel, there are a lot more cilia in the perfused conditions than there are in the static conditions. And if we look at the SEM underneath, we can see there's a higher density of cilia within the perfused conditions compared to the static conditions, which suggests that there's an increased ciliogenesis when dynamic perfusion is applied. On this slide, we can see two videos which again show the improved ciliogenesis when the culture is cultured under dynamic conditions compared to in static conditions. So I'll just run the dynamic video first. So here you can see the cilia moving and the increased number of cilia compared to the static video on the right-hand side, where we don't have the same amount of cilia. So I'm just going to run that through for you one more time. Dynamic compared to static. The lung model under perfused conditions also show improved mucus production, and they showed this by staining for MUC-B. And in the panel on the left here, you can see that as the purple colour within the photographs. So under perfused conditions by day 7, compared to static conditions in day 7, we've got a vastly improved production of mucus. They also ran the experiment out to day 21 under static conditions. And here you can see that there is some production of mucus, but it's not comparable to what we saw at day 7 in perfused conditions. They also had a look at the barrier functions and saw that this was again speeded up and improved when the cells were cultured under perfused conditions. By day 7, by staining for occludin, we can see an increased formation of barrier function. If we compare that with static conditions, you can see it's a lot less under these. But then if we look at the static conditions at day 21, it's comparable to those seen under perfused at day 7. This suggests that by culturing the lung model under perfused conditions, we can fast track the development of an in vitro model. They also had a look at the barrier conditions by looking at the tear measurements. And here you can see for the two different cell types, so the bronchial on the top and the small airway underneath, that there is improved tear measurements in perfusion compared to static for both of these cell types after 6 days. So what we're seeing here is not only a fast-track development of the lung model, but also improved mucus production and better barrier function and improved ciliogenesis. So we looked at the lung model first because that's the most recent paper, but the data that we have the most for the Quasi Vivo system is in the liver. So I'm going to move on to the liver now. So this experiment was done in conjunction with Sanofi in France, and basically we wanted to evaluate the use of the system for primary human hepatocyte culture. We decided to focus on the gene expression of a number of different CYP genes. You can see the example of CYP3A4 here. So this experiment was designed so that after we've either obtained the primary human hepatocytes from a supplier or isolated them themselves, we would want to hold them for up to 7 days in static culture. So this gives you 7 days to get the experiment ready and design your experiment and your assays. We looked at the mRNA expression of all these genes in freshly isolated hepatocytes. And this is obviously a relative scale, but the freshly isolated heps had a relative expression of this gene of around 20. We left them in static culture for 7 days. As you can see, we completely lose expression of this particular gene. At day 0, we either continued to culture them in static or we turned on the flow in the Quasi Vivo system. I think it's quite clear here that the static culture in our hands has completely destroyed the functionality of this gene. However, after 2 days, we've recovered around half of the gene expression, and after 4 or 5 days, we've completely recovered the function of this particular gene. So the flow conditions are restoring function of CYP3A4. We were also interested in a number of different genes in the liver, so across phase 1, phase 2, transporters, and transcription factors. And this graph has been normalized to the freshly isolated hepatocyte level, so that's at 0. And here we've got the dynamic conditions in blue and the static in red. And as you can see, for most of the genes that we looked at, dynamic flow was improving the gene expression of these genes in our hands, so in the Quasi Vivo system, compared to the static. So we've got significant downregulation in static conditions compared to in flow. Obviously, it's not enough just to look at the gene expression. We have to understand if the enzymes are actually being expressed and if they're working. So this was a dose response curve done with rat primary hepatocytes, and we exposed them to diclofenac and then did an IC50 assay. And here we have the static situation represented in red and flow in blue. So if we follow across the 50% viability of these cells, you can see in static conditions, it comes out about 550 micromolar. The flow predicts the IC50 at around 50 micromolar, and we have seen toxicity in the clinic at around 4.2. So in this situation, using rat primary hepatocytes, we've moved the IC50 much closer towards the problems that were seen within the clinic with this drug. We've also got some users at the University of Edinburgh who have been using their stem cell-derived hepatocyte-like cells in our Quasi Vivo system. So in panel A at the top here, we can see the expression of CYP1A2 protein in static conditions and then two different flow conditions. So we can see here that the protein expression is increased significantly under flow. They also looked at the bottom panel on cell viability. So here they used a BMS compound which requires CYP2D6 activity to convert it into a toxic endpoint. So if the liver and if this enzyme in particular is functioning, it will turn it into a toxic metabolite and the cells will die. And what they showed in this experiment is that the compound had a detrimental effect on the viability of these hepatocyte-like cells, which means that this enzyme is definitely working. And we can see a significant decrease in viability by 5 and 9 times in response to flow. We've also been working with upside. So these are a cell line, and the same sort of experiments were done. So we looked at, these authors looked at primary human hepatocyte expression of 3 CYP proteins, and they also looked at this liver organoid under flow. So here you can see that all 3 of these CYP proteins have an increased expression in the Quasi Vivo system compared to the freshly isolated primary hepatocytes. On the right-hand side of this slide, you can also see that there are comparable architecture between the liver organoid on the left and the human liver on the right. We're starting to see the same sort of things appearing in the organoid as we see in the human liver. Okay, moving on to cardiovascular. We recently concluded a project with Manchester Metropolitan University here in the UK, and we were looking at creating a cardiovascular model. And we wanted to only use human cells in this model. So we've got primary human cardiomyocytes, smooth muscle cells, and endothelial cells cultured in one chamber on a 3D scaffold. And after we'd shown that you can do this, you can culture them in 3D under flow, and they do survive for 7 days, we exposed this model to doxorubicin, which is a cancer drug that's been shown to be toxic in some situations in the clinic. And here you can see that all 4 conditions, so scaffold under flow, cover slip under flow, so 3D versus 2D flow, and then scaffold static and cover slip static, all showed a slightly different profile when we looked at the percentage viability of the cells after exposure to this compound. So here you can see, for example, the cover slip static situation is vastly different from the scaffold under flow. We also have some users who are looking at generating a 3D cardiac biosubstitute using the Quasi Vivo system and some highly porous biocompatible gelatin scaffolds. So I'm just going to take you through how they created this model. So first of all, they seeded human mesenchymal stem cells onto their gelatin scaffolds to provide interconnected vessel-like structures. Secondly, they had human cardiomyocyte progenitor cells that had been previously stimulated in vitro to obtain their commitment towards the cardiac phenotype, and these were added into the culture. And then they put the whole thing into the bioreactor, which allows for perfusion of the whole scaffold. And their hypothesis was that this would provide superior performance in terms of cardiac tissue maturation and cell survival. So there's lots of nice results in this paper. I'm just going to run you through 3 of the graphs that they present. So if we look at the top left-hand graph, this was a measurement of DNA concentration within the bioreactor. so static versus dynamic. And dynamic conditions do provide a higher increase in scaffold cell colonization, but it's not significant when we compare it to the static situation. So in the top right-hand graph, we can see that it is more the change from the 2D situation into the 3D situation that is increasing the number of differentiated cardiomyocytes, as evidenced by FACS analysis. So here we're showing that it's really the 3D environment that's providing them with the biggest benefit. However, when they looked at qPCR of specific cardiac genes in these cells in dynamic conditions compared to static, we can see that in dynamic conditions, this is the bottom graph, compared to static at 0, we're getting a much higher expression of these specific genes. So we think that the combination of flow and a 3D environment is providing these cells with a better environment, so they behave more like the cell types they're meant to behave like. And finally, we're going to take a quick look at co-culture. So the first one I'm going to introduce is the blood-brain barrier. So this was, again, an Innovate UK project that we ran with University of Leeds and Queen Mary in London. So this was the ultimate aim of the project. We wanted a triculture of human, again, primary astrocytes, pericytes, and brain endothelial cells. These would be connected to 2 different pump heads with a insert inside the QV600, which is shown in the diagram here. On the top of the insert and exposed to flow in the top of the chamber, we've got the endothelial cells. On the base of the insert, we've got the pericytes, and then in the base of the chamber, we've got the astrocytes, and these are both exposed to flow being provided in the bottom half of the diagram. The ultimate aim of this model was to have a look at passage of certain compounds across the barrier to see if we could predict whether they would be able to pass from the blood flow into the brain. We didn't get quite that far, it was quite a short project, but I can share with you some data that we obtained. So this is looking at the viability of pericytes and the endothelial cells in 3 different situations. So the first situation is the standard media that these cells are supposed to be cultured in, and that's in the black bars. The second thing that we tested was conditioned media. So this is where cells were grown in their specific media beforehand, and then they were switched into the flow model with some of this conditioned media exposed to the cells. And you can see that the pericytes really liked the conditioned media, so we saw an increase in the viability of these cells when they were cultured in conditioned media compared to in their standard media. And the third situation was putting the cell types in the conditioned media in our flow system. And if you look on the right-hand side panel, you can see the endothelial cells didn't really mind whether they were in their normal media or in the conditioned media, but they really enjoyed it when we switched on the flow. So by combining flow in the Quasi Vivo system with the conditioned media, we're improving the viability of both of these cell types. There's also a published diabetes model. So this was done by the original inventors of the technology at the University of Pisa. So in this diagram on the left-hand side, we've got 2 MCMB modules, which is the QV500, and we've got one kind of flatter cell culture chamber. This different geometry cell culture chamber is designed to culture endothelial cells, which really thrive in the higher shear stress that this kind of cell culture chamber can provide. The 2 QV500 modules were used to culture hepatocytes in one and adipocytes in the other. These 3 cell types are all within a common media, they're all in the same flow circuit, and they're talking to each other. Basically, what the authors of this paper showed is that the triculture is far superior to the double culture at maintaining homeostasis. So if we take the top left-hand diagram, for example, which is measuring glucose, over 48 hours, we saw a decrease in the amount of glucose in the system in the double culture, but not in the triculture. And if we look at the top right-hand graph, we can see that the double culture, we saw an increase in FFA at the beginning of the experiment, which maintained throughout the 48 hours, and we don't see this increase in the triculture. So that's our diabetes model. And where are we going? So this is the main sort of activity that I'm focusing on at the moment as R&D manager at Kirkstall. So can we start building more complicated co-culture models? So the system is really ideal for these sort of connected cultures where we can culture different cell types and different chambers, and we can start thinking, how are they affecting each other? And by having the two different cell types or the two different representative organs together, are we then generating a better in vitro model? So I've got two possibilities to share with you today. So one is our gut model liver cell model. So here you can see the gut model is in the QV600. We've got two completely separate halves of the chamber with the gut cells cultured in the middle. The top half represents the gut, and obviously in an ideal situation you would have representative bacteria within this media. And the bottom half represents the blood circuit. So we can look here at any ingestion exposure through the gut layer. Downstream of the gut model, we've got our standard QV500 module with a hepatocyte culture within it, and these are connected and talking to each other. And along the system as well, you can see that we can integrate very easily sampling ports, because they're basically injection ports, and because they're powered by Lurlocks, as is our system, we can have these sampling ports at various points throughout the circuit to either inject or to pull out media for sampling, for example. So we could sample the media before the gut model and after the gut model. This might tell us if the compound has actually got across the layer. And then we can sample before the 500 chamber and after. And here we can see if the hepatocyte cell culture is modifying the compound in any way. So by connecting these two cell cultures together, hopefully we're providing a more complicated, but more kind of representative model. The other example that we could think about is a respiratory model. So in the same sort of idea, we've got the QV600 acting as an exposure module with a respiratory model cultured on a transwell. Here we've got the air circuit. So basically what we do is we loop the top half of the QV600 back on itself so that it's exposed to air, and the silicon is permeable to air. So whatever environment the chamber is in, that's what the cells will be exposed to. On the bottom then, we've got a blood circuit, as in the previous example, but here we might have a cardiac model, so a heart model downstream. So we can see if anything being inhaled is A, affecting the respiratory model, B, passing across the respiratory barrier, and then C, getting downstream to any of the target tissues that we might think that inhaled compounds might target. If you wanted to make this even more complicated and therefore even more representative, you might want to put things like a neuro model in there as well, and perhaps a liver and a kidney model. So that's the kind of direction that Kirkstall is going in at the moment. And here's just an example of what this would look like using the Quasi Vivo system. So here we've got the two-headed pump, and you can see the reservoir bottle here which holds the media. The reservoir bottles that I forgot to mention have a filter on them. This is to ensure sterility of the system, while also making sure that there is no pressure builder within the system. This reservoir bottle is then feeding media into the QV600, which has a transwell in it with the respiratory model. And the base of this chamber is then talking to cells in two downstream QV500, which could contain, for example, a liver model and a cardio model. And the whole system is recirculating. So that's the kind of direction that Kirkstall are hoping to take the Quasi Vivo system in the future. So in summary, we have the ability to closely control shear stress. That means we can fine-tune the flow rates and the chambers for a number of different cell types. It's really easy to use. So we've got protocols, which means that postdocs, PhD students, people in industry can run the system without any training. But we also do provide trainings as well. If people wanted a Kirkstall member of staff to come in and show you how to use the system, we can do that too. It's not to scale, so we get enough cell material here for assays such as qPCR and Western blotting. Even though the system is smaller than some static cultures, the cells are more metabolically active and more healthy. So we do think that you're getting more cell material, even though it might be smaller than a 6-well plate, for example. I didn't mention non-specific binding, but I'm going to mention it super quickly now. So the whole Quasi Vivo system has a low surface area to volume ratio. So if you're worried about any compounds or proteins or anything that you're putting within the system, If you're worried about them binding to the plastics rather than to your cells, our system is superior to static culture. If you're really, really worried about it, then you might want to look at the QV900. This has been manufactured from acrylic, as I mentioned, and we've shown in collaboration with some users in the US that if you combine the 900 with Teflon tubing, we get negligible compound sticking to the plastics. So you can add compounds into the system and nothing sticks to the plastic. Finally, you can do co-culture of multiple cell types and organs, which leads to more systemic models. Here is a list of some of our recently published papers. I'm not going to go through all of them, but you can see a list on our website, which is www.kirkstall.com. And that's me done. It's time for your questions now. So I'm going to.
Hello, I'm Jenny, and we're going to start the question and answer session now. So we have quite a few questions, and we're not going to have time to go through all of them, but we will email, if you have asked a question, we'll email you, ask her, and provide an explanation or an answer for you. So the first question, do you get the same results if the chambers are closer or further away from the pump?
Okay. When you set up the system, we do recommend that you calibrate it with the number of chambers that you're going to have in your experiment. You would also calibrate using exactly the same amount of tubing that you would be having in your experiment. So I think as long as you were calibrating, if you had an experiment that was close to the pump or further from it, you would negate any of those questions. In the small distances that we're using, so for example, in the picture of the 900 and on the 6-channel pump, some of the plates are closer to the tube, to the pump head than others. We don't think this provides a significant change in flow rates, but I would say that if, for example, if your chambers are closer to the pump, that probably means you're using less tubing than if the chambers are further away from the pump. I would calibrate both of those situations just to double check that the flow rates you're experiencing are the same. We've done a number of different calibrations with different numbers of chambers and tubing lengths, and the flow rates don't change, you know, if you increase the tubing. So I would say you would get the same results if it's closer to the pump or further from it. The thing that's more important than distance to think about is the height of the pump in relation to the chambers. So the pump that we have and the whole system itself will fit within the incubator. So that solves any problems there because the pump and the cell chambers are very close to each other. If, for example, you're using a pump that has to sit outside of the incubator, if it's much higher or much lower than your cell culture, then you'll have some kind of gravity problems, and you know it might be a little bit different. So I would just say as long as you're calibrating in each situation, that's absolutely fine.
Okay, so our next question is, do you experience problems with bubbles in the system, and how do you overcome or prevent this?
Okay, so as I suggested in one of the early slides, the system was designed so that you don't get any bubbles. If you remember, the inlet has a smaller diameter than the outlet. The roof is also sloped and it's peaked. So if you do get any bubbles in the system and they get within the chamber, they will just get shepherded out through the tubing and then eventually into the reservoir bottle, where obviously they disappear. If you do start to get bubbles in our system, that's generally indicative that the tubing in the pump head is starting to wear a little bit. That's really the only part of the Quasi Vivo system that needs to be changed on a regular basis. So if you are seeing in our system bubbles, that generally means the tubing's wearing down. But in most situations in our system, we don't get bubbles because the original inventors designed it very, very carefully to ensure that you didn't get any interruptions or bubbles in the system.
Okay. Are there any differences between the two channels on the same pump?
So again, as long as you calibrate both the channels before you use them, that will give you confidence that they're the same. The channels on the two-head pump are independently controlled. So if you wanted them at the same flow rate, you would just set them to the same dial number. We're fairly confident that the dial numbers on the two controls on the two-head pump are exactly the same. So if you have them set, for example, at 2, then that provides the cell culture with 200 microliters per minute cell culture. Again, I'm going to say it again because I think it's really important. You just need to calibrate your experiment, a couple of times, obviously before you start, and then maybe after you've used it a few times, just to be 100% sure for yourself in your brain that the flow rates are the same. But there are no differences between the two channels on our pump.
Great, thank you. Do you get any problems with contamination if you run an experiment for several days?
Okay, so the system was designed to be set up within the cell culture hood. So if you use general aseptic technique that you would do for static cell culture, you shouldn't have any problems. If you're using the correct kind of filter on the reservoir bottle, so that's the blue 0.22 micron filter, that ensures you don't get any contamination from the environment. I mentioned that you do have to swap out the media. This is generally done by emptying some from the reservoir bottle and topping it back up. Again, this is done within the flow hood, so as long as you're following standard aseptic technique, you shouldn't get any contamination within the chamber. The silicon is all, tight to microbial contamination, so you shouldn't get anything coming in through the tubing or the chambers themselves. The chambers, so the QV500 was designed with a really good locking mechanism with the base and the top of the chamber, and the lids on the 900 and the 600 were also designed to be tight, so you won't get any contamination through there. Again, the final place where contamination could creep in would be the lure locks. These are used to connect the chambers to each other and the chambers to the reservoir bottle. As long as these are done up really tightly every time, you won't get any contamination within our system. So we don't see it as a problem.
Okay, next question. So one of the viewers has noticed that during the introduction, you specified the use of Quasi Vivo is mainly to mimic interstitial fluid flow rather than blood fluid flow. Have you got any new evidence from modelling software showing that we can't reach these physiological flow rates?
Okay, that's a really good question, and it's an application that we're currently working on at Kirkstall. So if you remember the diagram that I showed of the QV500, the flow up to 1 ml per minute is really nice and laminar within the chamber, and the cells cultured at the base are experiencing these more interstitial flow rates and physiologically relevant levels of oxygen for hepatocytes. If you imagine, for example, moving the base of the chamber up towards the flow, you're going to pull them more into the flow, if you like, and the shear stress is going to increase. So we already have some inserts available for the QV900, and we're working on a variety of different inserts, which means that you can raise your cell culture from the base of the chamber into the flow, which should mean that they're exposed to higher shear stress, so starting to mimic the more vascular, if you like, flow. This also means that because we're using inserts rather than changing the geometry of the chamber, you can have different chambers with different shear stresses and different oxygen concentrations. So for example, if you wanted to connect a cardiovascular tube with, I don't know, a liver, you could have the liver cultured in a normal chamber and then the cardiovascular representative tube in a chamber where the insert had been used to raise the base. So you're using the same flow rate, the same circuit, the same media, but you're providing them with different shear stresses just by raising the base of the chamber.
Okay, so the next question is sort of joining a few questions together. What flow rates do you recommend for different cell types?
So every cell type is different, depending on where they come from in the body, what their requirements are. We do have a bank of knowledge from the cell types that we have experience with that we can share with you. So if you've got any specific cell types that you're interested in, please email me. My email address is on the screen, and I can see if that's within our bank of data. We have also a sort of sweet spot that most cell types seem to like, and we can suggest 1 or 2 or 3 different flow rates that we recommend that you would start with, for example, and then you could very quickly narrow down the types of cells, the type of flow rates you would need for different cells. Just to give you a little quick example, like the blood-brain barrier model that I showed earlier, they're very low flow rates. So within our system, they're really kind of pushing the limit of the pump. We're down at 50 microliters per minute. Human hepatocytes, so primary human heps, sort of 150 to 200 microliters per minute. If we're looking at things like kidney cells, that really are exposed to a lot of flow within the body, you can push them up to about 500 microliters per minute. But we do have a bank of knowledge and a network of users. So if you've got any specific questions, please let me know.
Great, thank you. So the next question is, have you cultured spheroids in the system?
We do have a number of users who have started to investigate spheroids. So both spheroids generated using some of the commercially available plates and systems, but also people who are making their own homemade spheroids. We do think that our system is really great for this kind of cell culture, because by providing flow, you're increasing the drive of oxygen nutrients into the center of the spheroid. So if you're getting kind of necrosis or problems of cell death within the spheroid, we're trying to negate those problems by using flow to drive oxygen nutrients within. We do have some data on liver spheroids, and we've shown that they do enjoy being in our system. One of the questions we had was, would they kind of float away into the flow? So would we lose the spheroids from the system? They don't seem to do that. The flow is gentle enough that they kind of just gather at the base of the chamber. So when you're applying the really gentle levels of flow, spheroid culture is improved in the system.
If the pump is in the incubator, will it have an effect on the temperature of the incubator?
It depends on the pump. So our pump, it shouldn't raise the temperature of the incubator too much. Also, if the incubator is self-regulating, then you should be able to negate that through the design of the incubator itself. We've had a number of users who have used the two-head pump now for 3 or 4 years. The only time people get problems with the two-head pump is if they leave it in a humid incubator while it's not turned on, basically. If the pump is on and it's turning and it's in an incubator, everything's absolutely fine. We had somebody who went over Christmas and left it in the incubator, and they came back and it didn't work. So don't do that. As long as it's functioning, it's turning, it's working within the incubator, both the incubator and the pump are absolutely fine.
Time for a couple more questions. Do you have any exposure models?
Again, something that we're working on, because we think that some of the in vitro models for skin and respiratory are really primed for our system. So if you're putting, trans wells with skin models or respiratory models within our QV600 chamber, you can then put them in maybe some commercially available exposure systems, so that we're spraying compounds or applying creams. So we are in conversation with a number of different companies who can provide these exposure models. We don't have them yet, but if you're interested, please email me, and I can share the kind of ideas and the people that we're working with to see if we can work together to create an exposure model.
What's the best endpoint assay to use with the system?
So the system was designed, as I said, for hepatocytes, but we think it's quite agnostic in that you can culture a number of different cell types. In the same vein, you can do a number of different endpoint assays. So if you read some of our papers, you can see that our users have done Western blots, qPCR, simple viability assays like MTT and LMR Blue. We can do immunohistochemistry in our system, with cells grown in our system. If you take the trans well out, you can start looking at TIA. So there are a number of different endpoint assays that you can do. Again, we try and be flexible so that whatever questions you want to ask of our system, we can answer it.
Okay, I think that's probably our last question. Again, we didn't manage to ask all of them, but we will email you with the particular questions. I know some of you had slightly longer questions that need a more in-depth answer, and Kelly will be in touch in the next couple of days. So I'll leave you to sign off now.
Great, thanks to everyone for attending the webinar. It's been a great pleasure to have you with us today. If you have any questions, please let me know. You might get some poll questions after the webinar from Simon, so please answer them if you've got the time, and hopefully we'll meet some of you guys in the future. Thank you very much.